Emily can hear this. So, actually, no, I sent you an email with a link to a Dropbox. Is that correct? Okay. Okay. Tomorrow is probably not the best idea.
I don't even know.
And she has a meeting with me in the morning anyway, so she's not free.
I'm assuming there's might be a lot of geese out if there's snow too.
Oh yeah,
Really? All right. I don't think it's going to take us all day to get through this paper because Avery is going to do a stellar job of leading us through it. And so let me just make sure that I put everybody's name in the list because I had to go and look up Emily's email because you guys have numbers in your emails now and you used to not.
Wait, what?
You have numbers in your emails? That's, I think, the year that you started. Yeah. So it's like I used to be able to just do last name, first initial, no problem. But now I have to remember what year you started at William Jewell College, which is beyond my brain's ability. Okay, it says there was a problem completing this request. All right. Let's do it this way then. Yeah, so you're Alexander M.18. Not helpful.
I think it's weird because like it's when you came here,
Yeah.
but like you transfer in. Do you get the year that you transferred in, or do you get like your class year?
I think it's the year that you actually get here.
Yeah, and that kind of fucks you up there. Here's the like your graduation year, but there's no promise you graduate. Then
There is no promise that you graduate. That would be a potentially false promise. Not a set in stone false promise, but a potentially false promise. All right. I've just sent you the goose data in an email. That might have been the smarter. Mail cannot be sent. Okay. Why am I being thwarted at every turn? All right, let's try this again. So how did it go? Were you able to find geese and all that? Good. Okay, good. So you didn't have to go out to Smithville? Good. I went down to the ballpark to give you guys a place that was a little closer to home. Good. Uh-huh. No, the geese, they're so used to being around people there. Yep, that's where I saw them last time I was there.
can we go to like the same places as them, or do we have to go somewhere?
Yeah, because the geese that they saw yesterday are not going to be there today, or if they are, they're interacting with different individuals, et cetera, et cetera. Yeah, not a problem. Yeah, that's nothing. There is nothing wrong with that. Uh-huh. Okay, why do I not have internet? It's not sending because for some reason don't have internet service.
Are the molecules in the, they're not inside your room?
No, they're up in lab. So I need to, or do you have pairs?
No, mine are in my dorm.
My house.
Okay. I was gonna say, you can. I got like 3 emails just now.
Oh, did you? Okay, all right. Sorry,
2 emails.
I don't know why it disconnected me from the internet. So let's pull that open. The goose data. Um, so I probably did because no, the internet was messing with my juju. So I hate it when the internet messes with my juju. So what I've got here is two data sheets within this workbook. Um, the first one is called focal animal, and so for this one, this is where you have your group size, you have the which animal in the group is it? Is it your first one, your second one, your third one, or your fourth one in your small group? And then this is just head up, standing still, head up, walking, head down, feeding. That should be head down, non-feeding, and sleeping. Let me go ahead and change that since there's a typo. Typos seem to be my rule. And so what I need from you is just the cumulative amount of time head up, standing still, cumulative amount of time head up, walking, rather than reporting each individual instances of it. I just need, of that 5 minutes, how much of the time was spent in that activity. May have been 4 bouts of 20 seconds each, but that's 80 seconds. The other thing is, report it in seconds, because that's going to be easier to report and easier to analyze than minutes and seconds. So that's for focal animal. And then for scan, once again, even though if you were in a large group and you may have had to have scanned that group 4 or 5 different times, just give me your cumulative counts in head up, standing still, head up, walking, head up, or head down, feeding, head down, walking. So I don't want individual subgroupings. Just group all your data together for your small group in one summary number. And if everybody's doing that, then that should be pretty easy to handle. And I just went ahead and filled in your names so that you could put it where it belongs, and that just helps me keep track of things as well. Elizabeth, you always come last because you're W. Sorry. So, like I said, I asked you to do 4 of each group size. How did your group sizes vary?
We had a pretty small block,
Uh-huh.
just like 5 or so, and then we had a pretty big one. I think my biggest one was
Okay. All right. So a good range of group size. All right. Good.
120. Yeah, like about.
It was a guesstimate.
Yeah, it's a guesstimate. What's you're up that high? You're definitely the large flock
You have,
category.
I didn't see, I wasn't really paying attention, but did you have like numbers for like small, large, small, medium, large?
No, I mean, just kind of wing it when you get out there. You'll see.
I mean, to be honest, I thought like large would be like 20, but you said 120. Yeah, large could be 20 if like your
Large could be 1,000.
If like your other classes maybe like 5 and 10, then a large class.
Yeah. So look at the variability that you have out there, and work with that. If your largest flock is 10, I would not consider that a particularly large flock. I would probably go someplace else and look for a bigger aggregation. 120, that certainly qualifies. So yeah, but they could be 1,000 easily.
Yeah,
I was.
we have at home 1,000, yeah, very rare.
Yeah.
Very common.
Yeah, I've been. So these are Canada geese that you guys are looking at, but I've seen aggregations of snow geese that would be in the thousands, sitting out in a field in the Missouri River bottoms, just hanging out, eating corn. And
surrounded by agricultural fields in my house, and there just be like geese, like it's
yep, yeah. And the thing about it is, when you get those large aggregations, one of the things that those large aggregations do is they attract predators. So there was one year when there was some stuff happening up in Canada with lemmings, there was a crash in the lemming population. So the snowy owls were migrating further south than they normally do in the winter. And so you were finding snowy owls all down in the lower 48, which normally you only find them up in Canada. And there was a report of a snowy owl over past Richmond. And so I headed out one day to see if I could locate the snowy owl and maybe get a photograph of a snowy owl. And while I was out there, I just kind of made a birding day of it. And I saw this big field of snow geese, maybe 1,000, maybe more snow geese in this field, basically eating the leftover grain that was dropped from the previous autumn's harvest of corn. And there were all these snow geese, and they're white with black portions of their wings. All these snow geese, and all around the snow geese, you see bald eagles flying around. And they're flying around waiting to pick off a snow goose. And you can also see some bald eagles hanging out in the fields. And when you got them in your binoculars, you could see that they were sitting on top of a carcass of a snow goose eating. And so these large aggregations attract predators. And so one of the things that people note is that with vigilance, as your group gets bigger, you can spend less time in vigilance because you have more individuals being vigilant for you, so you have more time to eat. But one of the trade-offs of that is that large aggregations might actually attract more predators, making vigilance more necessary. So there comes a point where you actually have to be more involved in vigilance, more than you would if you just took the total amount of time and divided it by group size. So it's a tricky thing to try and assess. So anyway, this is the spreadsheet, and just dump your data in there, and we will look at them tomorrow, assuming that if nothing else, I'll have your guys' data. The analysis, the procedure for the analysis is going to be the same. It's just that when we get your data in, there's going to be more data to look at. So I am working on that. I hope to hand them back tomorrow.
Speaking of tomorrow,
Yeah.
there's going to be a big snow, like, it's actually decided we're going to leave tonight
Uh-huh.
for Richmond,
Uh-huh.
and so I will not be in class at all.
I will be filming because I think Emily is not going to be there tomorrow. Is that the case? Anyway, I'll record it just like we do normally, and so I'll record it. Also post the R script on the Moodle site so that once the data are all in there, I'll post the R script and the data files so that everybody has access to the data, and you'll be privy to everything. I might not record it on there. I might actually just do a recording on my screen so that you can see the R script
Yeah.
better. But yeah, we
We
can
should
handle
decide
it.
that night, but I mean, I think nothing hurts those predicted 5 days.
Yeah, it's predicted to be kind of yucky tomorrow, so I'll be here because I can walk. If nothing else, I can walk the 9 blocks to school. So, but
Sounds kind of cold.
yeah, well, it happens.
I don't even want to walk up from the parking lot.
Okay, all right. Any other questions about that?
Do we have anything for tomorrow's lab?
Your brains. That's it.
Okay.
Your brain's something to write on, take notes with. You might want to bring your laptop if you have R loaded on it, just so you can kind of follow along. If it's just going to be the two of us, then I'll probably just sit at a desk with 2 of you sitting behind me so you can watch what I'm doing on the screen. If Emily is there also, then we'll probably just put all 3 of you behind me. You can just look over my shoulder and follow what I'm doing. All right, group discussion. Avery is the facilitator today. Avery's job is not to walk you through the paper. Her job is not to describe the paper to you, because presumably we all 3 have read the paper. Her job is to get us involved in a discussion of the paper so that we can be contributors to that discussion. So she's more of a guide or a facilitator. She's not a presenter per se. I'm going to behave as if I am a student. So take it away, Avery.
All right. I'm just gonna read the abstract, I guess. I don't really know what I'm gonna do. There's growing evidence that adaptation may relate to size conditions of an individual phenotype. Those previous studies have concentrated on discontinuous variation. We show here that the copula or copula? Does it matter?
Copula duration. So copulation is just the act of having sex.
They're both saying that, right? That's all.
Yeah, copula. You see the word copulation, you see the word in copula, in copula, like the Latin version of it. So that's what we're using.
Copula duration of male dung flies, a widely cited example of optimality in biology, is tunely precise and continuously to the size of the copulating male. During copulation, new sperm displaces previously stored sperm at a rate that shows exponentially diminishing returns. Thus, at the optimal copula duration, a male's paternity gains and continued copulation drop below that expected from searching for a new female. Small males have lower rates of sperm displacement. They also have less prospect of population, but take over females of other males. I'm thinking this was a bad idea. Both effects predict that small males will copulate for longer, although displacement differences exert much the stronger influence. Observed copula durations are negatively correlated with size and concur with the optimality model across the natural male size range. This represents one of the first quantitative demonstrations of optimal responses covariant with continuous aspects of phenotype. Phenotype limited optimality profiles pose interesting evolutionary questions. So we are looking at the optimality of dung flies kind of in relation to their duration of population. Dung flies, we talked about them a bit before. They arrive at fresh cattle droppings, kind of wait for females that are carrying eggs to arrive, and then they copulate. Let's see. For this study, they kind of did the marginal free return, and you can see that in that first graph with the gains compared to the searching and guarding time, as well as the optimal copula determination. I'm gonna be honest, I really don't know how to do this.
You don't know how to do this because you're in presentation mode. We've trained you over the years to
Yeah.
present, and we haven't trained you to lead discussion. So rather than reading the abstract, which was a terrible idea,
Yeah.
which you sensed partway through it, ask Mason, for example, what is a dung fly?
What is a dung fly, Mason?
Just a fly that takes advantage of that area to copulate and go about his instill genetic livelihood that he's supposed to do, which is copulate with females and ensure that his sperm is not displaced and does not get taken over, and so that way it becomes more fit, I guess.
So, Elizabeth, why would a male want to hang out at dung rather than hanging out in the grass adjacent to the dung, for example?
That's where the females are.
Because that's where the females are. Why are they coming to the dung?
I know it's where they lay their eggs.
It's exactly where they lay their eggs. So the males are hanging out at the dung. The females are coming there to lay eggs. Mason, why fresh dung rather than dung that's 3 days old?
Surface of the dung.
I asked Mason.
I was gonna say it's just a better spot for them to lay eggs. It's more, I guess, nutrient dense for them.
It's not as much nutrient dense, it's what?
The surface of the dung deteriorates.
It dries out over time. And so even though there are nutrients in there, they're harder for larvae to access, for example, simply because it's going from a nice, wet, googey mass of feces to a dry, crusty mass of feces, so it's more difficult to consume when it starts to dry out. Do you see what I'm doing?
Yeah, I just really don't know how to do that. But we're
So think about the things that you think are important about the paper, and rather than telling us what those are, ask them what those are. So they're looking at these dung flies from the perspective of the male's perspective. How long should the male stay in copula with a female? What's the kind of theoretical model that they're going to use to kind of interrogate that idea with? they using ideal free distribution? What are they using? It's in figure 1. That figure, when you see that figure, you should immediately think what?
Ideal free distribution.
That's not an ideal free distribution, it's the
It's showing the marginal value.
It's the marginal value theorem. So who can explain to us what the marginal value theorem is? Yeah.
So basically, just like in this example specifically, the males shouldn't keep copulating with the females if they're not like gaining anything from it. So if the males aren't displacing sperm at a high enough rate, or if they're like they've already displaced an opportune amount of sperm, or if the female doesn't have any more viable eggs, then they should stop copulating with her because they're just wasting their time.
Okay. And that so over time the displacement rate is still increasing, but it's increasing at a decreasing rate.
Yeah.
So there comes a point where they should give up, and that's influenced by
the gain they would get from going to another female.
yes, but that's determined by what?
The search time.
The search time, how long it takes them to find another female. Did you say that just now?
Yeah, I said the gain they would get from going to another female.
Okay, but
they have to find her.
They have to find her, so you have to factor in that search time. Avery, can you do this?
Yeah.
Are you sure?
No.
Because right. Here's the deal I'll cut with you. I'll do today. Pay close attention to what I'm doing today. How much effort did you put into preparing for today?
We were just reading it a lot.
Okay, so you can't just read it. You have to actually create a plan for what you're going to do and how you're going to do it. So the act of facilitating, I thought I had talked about this before when I facilitated
You probably did.
the Shrike paper, but let me model it again today. I'll let you off the hook for today. Mason was going to lead discussion on the Brody paper and the Brody and Jansen paper. Why don't that day we split that? You do Brody and Jansen, he'll do Brody. You can take another run at it. But today, pay attention to what I'm doing and how I'm doing it so that you learn. And Mason wasn't here for the time that I did it for the Shrike thing, although it's on video. So the goal of facilitating a discussion is to kind of know what the important points are that you want to make, that you feel are really necessary for a person to get out of the paper if they're going to understand the paper. And then the other thing that you have to figure out is how can you craft a set of questions and be prepared for follow-up questions if they're not quite getting what you wanted them to get out of the first question you asked, to draw that out of the people sitting in the room. Presumably we've all read the paper, so asking you guys questions shouldn't be a difficult thing because you've read the paper, you've thought about it, we've talked about the marginal value theorem in class. So really the act of facilitating is leading people through the paper while not telling them anything about the paper. So I'll take today, you'll take Brody and Jansen, you'll still get credit and a grade for facilitating, you just won't facilitate this paper. Awesome, okay. Let's continue. So the marginal value theorem, this idea that you get fertilization gains the longer you stay copulating with female, but those gains are increasing at a decreasing rate, and then the determination on when you need to leave is determined by how long it's going to take you to not only find another mate, but what's the other thing that the male has to do? Has to guard the mate while she's laying her eggs. So there's this combined time period of both searching for a mate, capturing her with her copulating, and guarding her that all factor into how long you need to copulate, because that is then affected by how long it's going to take you to find another female to mate with. So that's what figure 1 is. So that arrow that points down is the optimal copula duration time based on a given search time of mu. As you went through this paper, were you thrown by all the symbols and all the equations? Okay. Do you have a good strategy for how to deal with those kinds of things?
I highlight in my papers and like make notes in the margins.
Okay.
So I made a lot of notes of like what the symbols were, and like went back and like when I came across a symbol or like an equation that I was like, what? I like made sure to go back and find it and be like, okay, that is.
I will oftentimes make myself on a sheet of paper a little table. Every time I come across a symbol, I'll just write down the symbol and what it means. So that when I come across the beginning of two paragraphs, I don't have to go back and find the earlier paragraph where it was mentioned first. What is G? Utilization gains. One of the other nice things about writing things down is you tend to remember things that you write down more than if you're just reading them. So this also helps with your memory. So mu is the time taken to find and guard an incoming female. T star is the optimal time, and how those two things interact with one another is determined by the gain function. How quickly the gains in fertilization decrease as you spend time with the female. Those three things interact to determine what the copulation time is. What did they find different than what had been found before that they wanted to look at in this paper in terms of how aspects of the phenotype of the flies affect optimal duration time?
You mean about like the displacement, the difference in displacement rate, and then
So there's displacement rate. How does that displacement rate vary among flies?
size, it decreases with size.
Okay, so displacement rate decreases with size. So what that means, in a sense, is large males have a faster displacement rate, right? The degree to which your sperm take the place of another fly sperm that's mated with that female before you is faster for large males. Small males, it is slower. So if that's the case, without looking at the paper, Mason, what does that mean for the marginal value theorem? If this is the gains, this is the displacement rate for large males, this is the displacement rate for small males, the gains, that's what the displacement rate is. The rate is actually how many gains do you make per unit time, and time is on that x-axis. If the time for searching and guarding is the same for both large and small male flies, what does this difference in displacement rate do to the optimal time that a male should be in copula?
Or it changes for the large and small.
Congruently. See, I'm even making people go to the board and draw stuff because I'm an ogre.
My line's not very straight.
No, that's fine. Just do your best. That's pretty close. So then what would optimal time be for the large male? Draw the vertical line.
Oh, yeah.
Now what would the result be for the small males with slower displacement rate? And it'd be up a little.
Yeah.
Just go ahead. Just keep drawing it and put it about where you would expect it to be. Have you drawn it cleaner? Yeah, so for small males, you have to spend more time in copula simply because it takes more time for you to displace the sperm that are there from the preceding male. Yeah, good. So that's what is in figure 1b, is these two gain curves. And what they haven't shown you is the other half of the drawing, the figure, the model. So this figure here is this half of this figure here, and then Mason's drawn the other half. So So size matters. Bigger males do a better job at fertilizing eggs because they have a faster displacement rate. That displacement rate then affects optimal copulation duration time, even if time for searching for and guarding a mate is the same between large and small males. Elizabeth, is the time T for searching and guarding males, is that the same for large and small males?
It's assumed to be yes.
It is assumed to be. Do they assume it to be in this paper?
I believe so. I thought they said they assumed it was the same. It says if all males experience the same mean time needed to find and guard a female, the smaller males should copulate longer than the larger males.
That is true. They go on and they write. Where is that exactly?
Underneath figure 1B, in the next, in that like paragraph.
That is the gigantic caption.
No, in the paragraph underneath that.
Oh yeah. Okay.
It's like
Oh yeah. Large males experience different. All males experience the same mean time to find and guard a female. That smaller males should copulate longer than large males because the tangent line was just value of T. That's what we just did. And that actually isn't T. I mislabeled this. This is actually mu in there. But that's what they say there. And it has an if in front of it, right?
yeah.
So if that's true, then this is true.
I think you go on to use the takeovers to prove that their time is shorter.
So Elizabeth, since I pitched the question to you first,
I thought that was kind of like a condition of like this is a time when that might be different and how it affects large males differently because they generally win the takeovers and it takes down their time by about half.
yes, exactly. So large and small males differ. They differ in displacement rate. They also differ in their ability to take over populations. The effect of that is that this is not their mu. Avery, what would the effect of takeovers do to large male? Time spent searching and guarding.
Lower it.
Shortens it, right? Yeah. So what effect does that have? Draw. See, that's the thing when you're a facilitator, you can just tell people to do stuff and have to. Yeah, sure. It's about half, right? That's what Elizabeth said, about half. Yep. Sure, run a straight edge. It's fine. It's fine. Okay, good. So this then is time corrected for takeovers, because this is mu for small males, this is mu for large males. This is then T star for large males. This is T star for small males. Because size makes a difference. Size makes a difference for two reasons. Displacement rate and affecting time required for searching and guarding. What can you assume about small males? Do they get to partake of takeovers? Yeah, they don't. They lose. When they try, they lose, mainly because of this disparity in size. Small male tries to take over a copulation from the big male. The big male is able to fight that off, stay on the female. And so you can assume that takeovers don't affect small males at all, but they do affect large males. Because when they attempt to take over, they succeed a certain proportion of the time. That then reduces the time that they have to spend searching. Now, what's one of the challenges of taking over a female? she's already been copulating with the male. You know for a fact that she's been copulating with the male. She's no longer a virgin. So all of her eggs aren't available. And so displacement rate really matters. So this is the gain rate, but we were talking earlier about displacement. Displacement rate is the rate at which you displace the sperm of another male that's already mated with that female. So that's different actually than gain rate, and I didn't really specify that earlier when we were doing this. But do they have a graph of displacement rate? Avery,
So what?
do they have a graph of displacement rate?
Figure 2.
Figure 2. What does figure 2 show us?
Well, they have the hind tibia length, which is basically just correlated to size.
Yep.
And then as they get bigger, the displacement rate increases. So larger males have a higher displacement rate.
So that's basically showing that part of what leads to that gain curve is the fact that males displace. You're a bigger male, you have more sperm. You have more sperm to put into the female, you get at a faster rate, and you as a result displace the sperm of the males that had mated with you previously. Is there anything else I'm missing up through the end of figure 2? So figure 3 is all about who gets to mate and who gets to mate via which avenue. So another way that you can think about papers like this is to kind of draw a path diagram, maybe. So you're a male fly. And you're on a dung heap. You have two paths to reproductive success, right? You have a female fly. She has eyelashes because she's female. She flies in and mates with the male. She may have already mated someplace else, or she may actually be a virgin fly, and either way, it doesn't matter to this male. Once she is mating with that male, this male will now guard her until she lays her eggs. While he's doing that, another path to reproductive success is this male taking over that mating and booting this male off and taking over. So this would represent a takeover. This would basically be an arrival. So this is the search time that is required. This is determined by how often females are coming to the dung patch. This represents the effect of takeovers. Both of these things are time. So these are all represented in time. Search time is assumed in this case to be constant, right? But takeover time is determined by size, more particularly your relative size relative versus the fly that you're trying to take over. So search time is relatively constant. Takeover probability this time varies by phenotype. The effect is small males, there's really no effect. Large males, time goes down. Okay? And that's what accounts for this thing. So what does figure 3 tell us? So figure 3 is, I think, one of the most confusing, well, second most confusing part of the whole paper. Mason, tell us about figure 3A. Explain to us what's going on there.
And the occurrence percentage versus the size.
Okay, so size. This HTL is the hind tibia length cubed. And what are they trying to get across to us there?
Unimodal line, and that basically size, the bigger or smaller doesn't vary, but I'm trying to say it's insignificant in a way.
They don't ever really do a test, do they? Could you do a test on this, Elizabeth? Test to see if these two distributions are different? Okay, you could. What kind of test would you do? I'm now asking about her about something that's from another class that I
Well, since
mentioned.
there's two categories, would you need to do a chi-square test of independence?
Maybe not a chi-square test of independence, but you might do a chi-square test of fitness of fit to see if the paired males, which are the black bars, is the same distribution as the unpaired males. So the black bars are paired and the open bars are searching. How did you guys handle? Did you guys do anything to that for to try and get yourself to figure out what's going on?
I just kind of took it as the small males had like they were searching more than the larger males.
Small males
paired.
appear to be, at least to me, to be searching more than the larger males, which tend to be paired more. So
What is the curve?
it's just the percentage. It's a frequency. It's just of the total, what percentage was in this category, this other category. So it would be hard to extract the raw data out of this and do a chi-square test, but one of the things that I like about having an iPad, I don't think iPads are particularly useful, but one of the things that they're super useful for is reading literature. And so I do stuff like this. Let's pull this up. So what I do, once again, this is just a helpful, a useful thing to do whenever you're working on a paper. I tend to underline things that I think are important, but I also tend to annotate my figures. So remind myself that G is fertilization gains. This is the line for large, this is the line for small, et cetera, because these captions are so long, I'll also circle where in the caption stuff for figure B starts, so I can go back and find it in the future if I need to. But in particular on this one, I have to remind myself that this is Heinz-Tibia length, that's their measure of size. So I write that in there just to give me a reminder, that thing means size. And then I do stuff like this. If you draw different color lines connecting, the red is paired, and the blue is searching, you can see that the peak for this is a little lower than the peak for this, which is a little higher, which indicates that maybe if we did a statistical test of this distribution, we might actually find that this distribution of searching individuals, the open one, is a little lower, because of course what I didn't do the final line, which is there are no searching males in this largest size category. There are very few large males that are in the searching. Sorry, there are very few small males that are paired. So the fact that you don't have any searching that are large and you have very few paired that are small, once again contributes to this shift in these two distributions. It's not a huge shift, it's a subtle shift, but it might be statistically significant. But the real question is it biologically significant? And as they talk about, that tends to be the case, actually. Why are searching males, why are smaller males, why do they tend to be more in the searching category than they are in the paired category? They can't perform takeovers, and they tend to, well, that would then just make them paired more often.
Didn't it say if they searched in the surrounding grasslands?
Oh, that's something different.
Oh.
Keep that in the back of your head. We'll get to that in a few minutes. That has to do with sperm replacement. They fail at taking over other populations, which means they are also being taken over more often. So two things contribute to them being in the searching category rather than the paired category. The paired males tend to be larger because larger males one, they're more successful at taking over, and two, they don't get taken over. So good reasons why this distribution might be slightly different. It's harder to see the difference if you just leave the graph alone. So don't be afraid to draw things on graphs other than labels and stuff like this. They're talking about distributions here. Just draw the distribution. It's easy enough to do, and it ends up making more sense to you when you've done that slight little modification. Avery, explain to us 3B, another kind of confusing graph because it's graphing two things on the same graph. So you notice that there are two y-axes. So tell us what this graph is trying to communicate to us.
The relative number of takeovers by size. So as they size increase, the relative takeovers also increase,
Okay.
and the searching time decrease because they were taking over.
So the open circles are about takeovers, the closed circles are about time. Small individuals are terrible at taking over. Large individuals are very good at taking over. And the effect of that on time to garden search is that small males spend a ton of time. Time is over here. What was it, 156, something like that? 156. That corresponds. So another thing that you should do when you read these papers is if they calculated that the time for taking over is 156, or sorry, the time to search and guard is 156 minutes, do they then show you a graph where they have that time? Does that time actually correspond to what is in the graph? And it does in this case. And so, and then it lowered to like 84, I think, for large males. And once again, that's approximately where this is. So size affects time to garden search, such that the smaller you are, the more time it takes. The larger you are, the less time. Part of that, at least, is due to takeovers. Confusing when you graph two separate variables on the same graph. And so you have to pay special attention to what's in the graph and which points correspond to which y-axis in this case. The open circles correspond to the left y-axis. The closed circles correspond to the right y-axis. So they did all of this. They found that large males should, in theory, spend less time in copula because they have higher rates of displacement and they have lower rates of takeovers. They don't get taken over as much, and they tend to take over more often. So that reduces the time they spend searching, which then reduces the time in copula. So they're interested in figure 4a because 4a is what they actually observed as size goes up, copula duration goes down. And it goes down in kind of a nonlinear sort of way. So what are they doing in 4B and C? And these are the two most confusing graphs in the paper in my mind.
The dotted-ish lines are that kind of what we just saw in like the A part, and
Yep.
then the
Exactly. So these three lines, this is the average. So this is the regression line, and this is the confidence interval. Notice how this y-axis is more blown up than this y-axis is, because what they've done is they've zeroed in on this section of the graph. So they go from 0 to 100 rather than 0 to 200, because you have these outliers. Some small males that spent a lot of time in copula. So they blocked that out because they want to concentrate on this section, which is just the prediction of the main bulk of the data. So you have these two effects that affect copula duration And you can mathematically model them, and that's what is in the caption to figure 1 is how you go about modeling all of this. In this case, they show what the prediction would be if only displacement rate was in the model and only if takeover rate was in the model. What does this tell us?
Displacement higher before defecting takeover said like nothing.
Yeah, displacement rate affects copulation duration way more than takeovers do. So takeovers contribute some to it, but it's not nearly as important an issue as displacement rate is. Elizabeth, what is figure C telling us?
I think basically just like the effect of both have.
The effect that both of them have. And what you see is that in this case, you get pretty good correspondence to of the model predictions to the actual observed data once you get to a certain size. Why do you not get good predictions over here on the left-hand side of this? Well, all flies are mature. So the fly life cycle is you start off as an egg, then you're a larva, then you're a pupa, and then you're an adult. So there was a short period of time, like right after they emerge from the pupal casing, but that time is really short. We find this in fruit flies. Males come out of the pupal case, and as soon as their wings are expanded and they're ready to fly, they're ready to mate. That's just kind of the nature of being a fly. They talk about this in the discussion why there's not very good agreement. Yeah, you're down at the extreme levels. These are very small flies, some of which have really long copulation duration. And so we can think of those as kind of outliers. If these guys over here weren't there, this whole curve would be much closer to a straight line than otherwise would be. And as a result, you would probably have a better fit of the model to the observed data. So if you were to summarize this paper in a couple of sentences for your grandmother who would think you were totally weird if you were telling her this, what would it be?
The population time based on the size varies on the ability of one's displacement. So size of the flies affects their displacement ability, which is the act of overtaking one sperm and another female.
Okay.
And because of that, larger males can do that quickly. That has the biggest effect on copulation time overall. So smaller males will copulate longer because of that. And then takeovers also play a role into that copulation time as well, but it wasn't proven as significant as displacement time.
Okay. Yeah, your grandmother thinks you're very weird.
Yeah.
So these males are out there copulating. They then also have to replenish their sperm. How quickly does that happen? Is that a time-consuming thing? Does the replenishment rate vary by size, according to these guys? And keep in mind, they don't show you any data on this. You have to kind of trust their word. But what's their claim about replenishing sperm?
Comparing in size.
I think they said it was like independent of size.
It's independent of size. So what's the really big difference between large and small males? So they both have to eat. Large males have to eat more. But what does the large male start off with?
The same amount of sperm?
They don't start off with the same amount of sperm.
Different amounts of sperm.
They start off with a larger amount of sperm because they're just generally bigger. They have bigger reservoirs to draw from. So you have to replenish your sperm reservoir, and this adds time to your activities because you have to feed, and because you're an adult, and this is true for a lot of flies. The dung pat is food for larvae. The dung pat is not food for the adults. And this is pretty common with lots of flies where the adult form of the fly, the adult life stage, feeds in a different way than the larval stage does. So they actually have to leave the dung pat, go off, and they feed on floral parts, nectar and things like that in the grass. And so that takes time away from the dung pat. But the advantage of while you're on the dung pat is you're a larger male, you have a larger reservoir, which basically means that you have more sperm to draw from, which is part of what probably contributes to your high displacement rate. So experiments show that larger males have higher reservoirs, but the proportionate rate filling is independent of size. So each of these large and small individuals will have to spend a certain amount of time away from the dung pat to replenish their sperm supplies, to feed, but the rate at which that happens doesn't vary by size. So here at the end, they talk about the evolution of this. Elizabeth, remind me what the 4 preconditions are for the theory of evolution via natural selection.
There has to be like differences in traits.
So there must be variation among individuals. There is variation among individuals. We have large individuals, small individuals.
It has to be heritable.
It has to be heritable.
Overproduction of
Yeah.
So not everybody gets to mate, and that's certainly the case here, or they don't get to survive, but in this case we're talking about mating. But what determines who gets to mate and how effective that mating is? Differential survival or differential reproductive success. And that is tied back to this variation in phenotype. So you have this phenotype, well, these 2 phenotypes. You have these 2 phenotypes, large and small. Large males, because you're large, there are a suite of other things that go along with being large. You have a larger reservoir of sperm. You have a faster displacement rate of other individual sperm in the female that you're copulating with. These 2 things lead to the fact that you can then have a shorter copulation duration, partly because contributing to that is your probability of takeovers, but a bigger part of that is actually due to displacement. What does that mean if you're spending less time with each female?
You can spend time with more females, you're spending time with less females.
You get to spend more time, you get to spend time with more females because you're spending less time with each female that you're with. So this suite of characteristics leads to potentially much higher fitness because of you're just big. Can you see that? Still can't read it because I wrote it too high on the phone. Whoa. Wasn't expecting that to happen. So, and then small is the same for all the rest of this. So in this last paragraph, they're trying to think about how this would evolve, because ultimately this is a decision that males are making. The male decides how long they're going to guard the female, how long they're going to copulate the female, et cetera. So what do they say about the evolution of this? What's the big trick? There are good evolutionary reasons why this might evolve. What's the stumbling block here? For thinking about this as a set of traits that can evolve.
The bond partners.
Okay, so what do you mean by that?
I think it comes with like Honestly, I kind of hard time understanding the last paragraph.
That's why I'm asking about it. So,
But I think it's talking about like how like things influence the phenotypes differently, so like they can't.
so one way of thinking about the evolution of a complex trait like this. So the phenotype here really is size. But that phenotype is then coupled with copulation duration. And so you have to envision a genetic system. So what they're talking about in the last paragraph is you have to envision a genetic system where there's a genetic system that is one of two types. One is that there's a gene for small males that then influences their displacement rate and their copulation duration and all that stuff. Then there's another gene for 15 HCl cube squared, or to the cube, flies that determines all of that for them. Another gene for 20, another gene for 25, another gene for 30, another gene for 35, another gene for 40, 45, and 50. So you have all these genes, but the gene only acts in one size of fly, and it's shut. All the other genes are shut off in that one size of fly. That's a pretty stretched out argument genetically to think that there's a bunch of genes. Each gene is only active in that particular size fly, and all the other genes are turned off for the lifespan of that fly. That's hard to grasp how that would work. The other alternative is that there's a more general set of genes that allow you to monitor, for example, what your sperm reservoir is. That allows you to monitor time, and that allows you to put those two things together cognitively to the point that you can assess how much time you should spend copulating with a female. Is there a gene for reading for humans? No. Yet reading is a phenotype that we all possess. We all have the capacity to read. But there isn't a gene for reading per se. What is it that we do have that allows us to have this ability to read?
I can't remember what it's called. Just a section in our brain that's responsible for it. So we just have cognitive ability.
We have cognitive ability, and we probably do have genes for brain size. We have genes for the organizational properties of our brains, how our brains are structured. We probably have genes for how neurons interconnect with one another. So we have probably a suite of genes that generally contribute to the level of intelligence that we do have. And what grows out of that level of intelligence is the ability to do a lot of things, reading, math, etc., etc. But there's not a specific gene for reading, there's not a specific gene for math, there's not a specific gene for abstract thinking. There's a set of genes that organize our brains, make our brains as big as they are, organize them in the manner that they are organized, make some part of our brains bigger than other part of our brains. You look at an animal like a reptile, their frontal lobe is much smaller relative to the total size of the brain than their hindbrain is. So we have a set of genes that determine that organization, those differences in size, etc., etc. And what comes out of that is our ability to do certain things. That's a much more likely scenario to see these traits co-evolving in concert with one another than to think about there's a set of genes, one gene gets turned on in one size fly, and all the other genes are turned off in that fly, etc., etc. So at the time that this was written, kind of thinking about these emergent properties of brains and things like that wasn't a very well-developed idea yet. It's something that came along later in science. But that's possibly what's going on. You have maybe a suite of a small number of genes, all that contribute to these flies' ability to self-monitor themselves, and also their ability to monitor their environment, and also the cognitive abilities to put those two pieces of monitoring together.
happy with the last paragraph? Okay, good. Do you see how this is done? It takes time to prepare for it. You can't just read the paper and feel like you're prepared. You have to read the paper, then you have to think about the paper. You have to think about the paper in a different way than you had to think about it just to write the summary. You have to think about the paper from the perspective of, I have to lead my people through this thing. I spent some time delivering information, but I spent a lot of my time drawing the information out of the participants of the discussion. Can you and Mason do that?
We'll try.
Okay.
I think so.
Right, I've modeled it twice now for you. So
I know how it's supposed to be done, but
It's a matter of doing it, honestly.
it's just
So-
difficult.
Yeah.
one of the things that you guys might want to do, since you're covering two different papers, but they're both kind of on the same topic, is you might just want to talk to one another about strategy.
Mm.
And then the other thing you want to do is you might want to just talk to me about strategy. I get paid to do this. I've been doing this a long time. My students hate that I ask so many questions of them, but that's how you get people involved in thinking about the thing that you want them to think about. So that's your goal. You want us to think more deeply about the thing that we've already read than we did by just reading it. Those of us not presenting have the lighter workload that day because we've read it at one level. Because you're facilitating, you have to have read it at a different level and be prepared to draw us into that deeper level. Questions? Oh gosh, we're out of time. I said it was going to not take us all period, but it took us all period. All right, you have literature summary for me. Awesome.
I can just run to the second floor and print mine.
Okay, yeah.
But I'm headed over for a research
Bring
meeting.
it to my office.
Yeah.
A little bit of grace, Avery.
Huh?
A little bit of grace.
What do you mean?
I gave you a little bit of grace today.
Thank you. Definitely would not have been asked for this.